Welcome to the Deep Dive, the show where we sift through the latest research, your articles, and our own notes to really pull out the most important nuggets of knowledge and insight.
Think of it as your shortcut to being genuinely well-informed.
Exactly, complete with surprising facts and, just enough humor to keep you hooked.
Hopefully.
Today, we're plunging into a topic that, well, at first blush, sounds utterly unbelievable, maybe even a little absurd.
It really does.
We're talking about the highly addictive drug cocaine, a substance so notorious in human society, and its surprising, profound connection to honeybees.
It's an incredible contrast, isn't it? Almost contradictory. On one hand, you've got cocaine as this chemical weapon,
Right.
a potent neurotoxin produced naturally by the coca plant.
Mhm.
It evolved over millennia specifically to protect itself.
How does it do that? By disrupting insects.
Yeah. By disrupting and deterring insects, messing with their systems. But then on the other hand, this exact same chemical is intensely rewarding, powerfully reinforcing, and tragically addictive for humans.
And that fundamental difference, that discrepancy, that's what scientists have long grappled with as the paradox of drug reward.
Precisely. It just seems weird.
And for the longest time, the traditional explanation, the seemingly straightforward one for this paradox, was pretty simple, wasn't it?
It seemed logical enough.
Basically, that there must be fundamental, unbridgeable differences in how mammalian brains, like ours, respond compared to invertebrate brains, like those of insects.
It just made intuitive sense, right?
Right.
If nature designed it to deter insects, it simply shouldn't be rewarding to them.
Case closed. Or, so everyone thought.
Exactly what everyone assumed. But our mission today is to dive deep into how a truly groundbreaking scientific paper from 2008,
Okay.
published in the Journal of Experimental Biology by Barron and colleagues, managed to meticulously challenge that long-held belief.
So this deep dive is focused on that one really rich source.
That's right. We're extracting the most important insights, and I promise you the conclusions are truly mind-bending. Get ready for some serious aha moments.
Because our unlikely protagonists today are...
None other than Apis mellifera, the common honeybee,
Yeah.
and their utterly incredible sophisticated dance language.
Wow. Okay.
We're going to unpack how this research meticulously pushed the boundaries of our understanding, revealing some truly surprising connections across the tree of life.
So let's unpack this grand puzzle. What was it that truly drove these researchers to investigate the effects of cocaine on honeybees? I mean, why bees? Why this study?
It all really goes back to that central paradox, that fundamental enigma of drug reward. We often think of cocaine primarily as a recreational drug, maybe a medical one, historically.
Right, in a human context.
But it's vital to remember its profound ecological role. As we touched on, it's a potent neurotoxin. It's made by coca plants, specifically Erythrocylum species, as a natural defense mechanism.
A plant defense.
Its purpose in the wild is quite literal, to critically disrupt insect motor control, basically stop bugs from eating the plant.
Ah, okay. So it protects the plant from herbivorous pests. And this isn't new knowledge.
Oh no, this defensive function has been well documented for decades. There was research back in the early 90s, Nathanson and his team detailing its insecticidal properties. It messes insects up badly.
Right, so it's a chemical shield, a natural deterrent, built by the plant to say, stay away or you'll regret it.
Pretty much.
But then, when you look at its effects on us humans, the story changes dramatically. Yes, it's highly toxic at medium to high doses, causes severe physiological damage.
Absolutely dangerous.
But at low doses, it's powerfully rewarding, intensely reinforcing, and, as we know all too well, devastatingly addictive.
And that's the core of the paradox. Why would a chemical that evolved specifically to deter and disrupt insects become such a powerful source of reward for mammals like us?
It feels almost counterintuitive to, how evolution should work.
Exactly. And as we just discussed, the prevailing common explanation for this paradox was simply that cocaine evolved specifically to be an insect deterrent, full stop.
Which implied a fundamental difference in brains.
Right, a categorical difference in how mammalian brains, especially ours, respond compared to arthropods. And for a long time, there seemed to be a lot of evidence supporting this view.
Like what kind of evidence?
Well, abundant data showing cocaine did indeed mess with insect motor systems. It caused disruptions, hyperactivity, uncontrolled movements, basically making them sick and dysfunctional.
Okay, so it clearly harmed them.
But, and this is the crucial missing piece that supported the old view, there was no previous demonstration of it being rewarding to insects. Zero evidence.
Ah, so the logic was harms insects, rewards mammals, therefore fundamentally different brain responses.
Precisely. On the surface, the paradox seemed neatly resolved to say, well, it affects them differently because their brains are fundamentally different.
Yeah.
Simple.
But then, here's where it gets really interesting, right? Where that tidy explanation starts to unravel.
This is where the plot thickens. New scientific data began to emerge that directly called this long-accepted solution into question.
How so?
Researchers started finding significant, really unexpected similarities in neurochemical systems. And critically, in cocaine's actual mode of action, how it works at a chemical level between insects and mammals.
So it wasn't as simple as different brains, different effects anymore.
Not at all.
Yeah.
The biological lines were definitely blurring.
What was the core similarity? What was this biological bridge they found?
It lies in what are called biogenic amine systems. These are ancient, fundamental chemical signaling pathways in the brain. Think neurotransmitters.
Okay, like dopamine.
Exactly like dopamine.
Mm-hmm.
And others too. In both mammals and insects, cocaine operates by blocking the reuptake transporters of these biogenic amines.
Okay, blocking reuptake transporters. What does that actually mean in, like, simple terms?
Right. Imagine these chemical messengers, neurotransmitters, being released from one nerve cell to signal to the next one across a tiny gap called a synapse. Normally, after they've delivered their message, they're quickly reabsorbed or reuptaken back into the first cell. It's like a cleanup crew clearing the signal so the next one can come through clearly.
Got it. Keeps things tidy.
Cocaine acts like a wrench thrown into that cleanup crew. It blocks the reuptake process.
So the messengers hang around longer.
Exactly. It leaves these crucial chemical messengers floating around in the synapse for much longer, continuing to stimulate the next nerve cell.
So essentially, it doesn't just send a signal, it cranks up the volume and keeps the signal going and going, like an amplifier.
That's a great analogy. It amplifies the existing signal by preventing the normal turnoff mechanism.
And which specific neurotransmitters are we talking about here, the ones getting amplified?
Well, primarily dopamine, DA, in mammals. This is incredibly well established. Dopamine is a key player in our reward processing, pleasure, motivation, even motor control. Its role is huge.
Right, the reward chemical we often hear about.
Kind of, though it's more complex than just that. But in insects, it's not just dopamine. It's also another related chemical called octopamine, or OA.
Octopamine, never heard of it.
It's often considered the insect equivalent or functional analog to norepinephrine in mammals. And it also plays a significant role in arousal, motivation, and reward in insects.
Okay, so dopamine and octopamine in insects.
Right. And what's absolutely crucial here, the thing that really sparked this research, is a profound functional parallel. These very same biogenic amine systems, the ones affected by cocaine, they modulate both motor control and reward processing.
Wait, in both groups, mammals and insects?
Yes, that's the key insight, this deep, conserved mechanistic overlap. The same systems doing double duty, controlling movement and mediating reward.
Wow.
That overlap was the scientific spark, the truly compelling reason that made these researchers revisit that fundamental question. Could cocaine actually be rewarding to insects, despite its natural role as an insecticide?
Could it, in effect, be hijacking their reward system in a way strikingly similar to ours, just because the underlying machinery is so similar?
That was the hypothesis. Could this plant defense mechanism inadvertently tap into the insect's reward circuitry?
So if you're a scientist wanting to test whether something is rewarding to a honeybee, I mean, a creature that doesn't exactly fill out a survey on how it feels,
Right, they can't tell you.
how do you even begin to measure something so subjective? This is where the honeybee dance language comes in, right, as this unique bioassay.
That's exactly it. Apis mellifera, our common honeybee, was an ideal, maybe even a perfect choice for this study, precisely because their dance language offers a natural, quantifiable, and incredibly well-understood way to study how they assess and communicate perceived reward.
It's amazing. When a forager bee finds a really good source of nectar or pollen, they come back to the hive.
And they don't just sit there. They perform these highly stereotyped, precise movements, these incredible dances.
To tell their sisters where the good stuff is.
Exactly. To signal the location. And crucially for this study, the value of those resources to their nestmates.
It's like a tiny, vibrating, living GPS and restaurant guide all rolled into one. It's such a complex, fascinating behavior.
Absolutely. For resources close to the hive, maybe within 50 meters or so, they perform what are called round dances. It's a specific pattern of movement, turning in circles, reversing direction.
Okay.
And here's the key. The likelihood that a bee will perform a round dance And even the rate or vigor of those dances, like how many circuits they complete per minute, are directly related to the value of the resources they collected.
So more dancing means better stuff.
Generally, yes. But this value isn't just about how sweet the nectar is, for instance. It's a complex, holistic assessment. It's almost like the bee's overall rating of the resource quality.
What goes into that rating?
It weighs many factors. The costs of the foraging trip, like the energy spent flying there and back, the actual benefits of the resource, like its sugar concentration or pollen quality, and even the current nutritional needs of the entire colony. Are they desperate for sugar right now?
So it's a sophisticated, integrated evaluation, not just a simple reflex.
Exactly. It's not just a motor response. It's a profound communication of perceived value. This system offers a unique, natural, and quantifiable way to assess a forager bee's perception of resource value. You can literally watch them and see how much they like what they found based on how enthusiastically they dance.
Which makes it perfect for testing if cocaine changes that perception.
Precisely. And that's what this study aimed to do.
Yeah.
Use the dance response to both sucrose, sugar water, and pollen feeders as a precise, measurable assay to assess cocaine's effects on honeybee reward processing.
They were looking for parallels with how mammals respond.
Yes. By applying cocaine in both acute, single doses and through more prolonged chronic low-dose treatments, they wanted to see if the bees would behave as if the cocaine made them perceive the resource as more valuable, almost as if they were shouting, "Wow, this nectar was even better than I thought."
Okay, so the why is crystal clear now. Unraveling a long-standing paradox about drug reward by looking at conserved neurobiology across species using this amazing bee dance. Now let's get into the how. How exactly did these ingenious researchers administer cocaine to bees? That seems tricky.
It required precision.
And what meticulously designed experiments did they devise to figure all this out? Because the scientific rigor here sounds truly impressive.
The experiments were conducted at the Australian National University in Canberra using standard, commercially available European hybrid honeybees, the kind many beekeepers use.
Okay.
The method of drug delivery was absolutely crucial for precision and control. They used topical application. A tiny, precisely measured droplet, just 1 microliter, which is incredibly small, a millionth of a liter of free-base cocaine, or other compounds like octopamine or meancerin, was dissolved in a specific solvent.
And they put this droplet where?
It was carefully applied to the dorsal thorax, which is essentially the upper back, of individually identified forager bees while they were busy feeding at a controlled feeder outside the hive.
And the solvent they used was called DMF, dimethylformamide. Why that one? Is there something special about it?
That's an excellent question. DMF was chosen specifically because it's known to penetrate the bee's cuticle, their waxy outer shell.
Ah, so it gets through their skin.
Exactly. It allows the dissolved compounds to enter their hemicole, which is basically their insect bloodstream.
Okay.
From there, the drug can circulate throughout their body and, importantly, reach the bee's brain. This was a critical detail to ensure the drug actually got into their system effectively and systemically, rather than just sitting on the surface. It allowed for a controlled internal dosage.
Makes sense. You need to know the drug is actually getting where it needs to go. So let's walk through the series of experiments they performed. Experiment 1 seems like the foundation, looking at cocaine, the dance, and that connection to biogenic amine signaling. What was the main question here?
The primary objective of Experiment 1 was really twofold. First, simply to see if cocaine did indeed affect dance behavior in the bees. Did it change how they danced?
Okay, the basic effect.
And second, if they did see an effect, did it seem to involve those crucial biogenic amines we talked about earlier? They really wanted to see if cocaine could alter how a bee communicates the value of a resource and maybe how it was doing that neurochemically.
And how did they set up this crucial observation it sounds like you'd need to see inside the hive?
They did. They used colonies of about 8,000 bees housed in specially designed glass-walled observation hives.
Very cool. Like an ant farm, but for bees.
Sort of, yeah. This unique setup allowed for direct, unobstructed observation of individually paint-marked forager bees. They could track specific bees.
So they'd mark them when they were feeding outside?
Yes. Mark them, apply the treatment, and then watch them when they return to the hive to see if they danced on the comb face.
And where were the bees getting their food?
These marked bees were trained to a feeder containing a pretty concentrated sugar solution, 1.5 mol per liter sucrose, positioned just 10 meters from the hive. Close enough for round dances.
Gotcha. And how did they collect the data?
Their round dances were meticulously video recorded over a 50-minute period after they returned. From these recordings, they extracted two key metrics. First, dance likelihood. What proportion of visits to the feeder actually resulted in a bee performing a dance?
Okay, did they dance or not?
Right. And second, dance rate. If they did dance, how vigorous was it? They measured this by counting the number of dance circuits completed per minute.
More circuits, more enthusiastic dance.
Exactly. And all observations began precisely 20 minutes after the bees received their topical treatment, giving the drug enough time to get into their system and take effect.
Okay, makes sense. And what were the specific treatments applied to the bees in this first experiment? You mentioned cocaine.
They used single, acute, topical doses of freebase cocaine at 3 different concentrations: 3 micrograms, 6 micrograms, and 12 micrograms, all dissolved in that tiny 1 microliter of DMF.
And controls. You always need good controls in science.
Absolutely essential. They had 2 crucial control groups. One was sham-treated bees. These bees were simply gently touched on the back with an empty microcapillary tube, mimicking the handling process but without any substance applied.
Controls for the stress of being handled.
Exactly. And the second control group got just the solvent, 1 microliter of DMF only. This was vital to isolate any potential effects of the solvent itself, ensuring that any observed changes were actually due to the cocaine and not just the DMF vehicle.
That's smart, covering all the bases. Did they also include a positive control? Something they already knew should affect dance behavior, just to check their setup was working.
They did, yes. That's a hallmark of good scientific design. They included a group of bees treated with 2 micrograms of octopamine, or OA, dissolved in DMF.
And OA was known to affect dancing.
Yes. OA is extremely well known and documented to modulate bee dance behavior, specifically increasing it. It's linked to reward signaling in bees. So it served as a crucial benchmark. They could compare cocaine's effects directly to a known modulator of bee reward.
Okay, so they could see if cocaine acted like OA.
Right, and then in a separate bee colony, they performed a really critical sub-experiment. They wanted to test if blocking biogenic amine signaling could prevent cocaine's effects.
How did they do that?
They used a biogenic amine antagonist called mianserin. An antagonist is something that blocks the receptor, preventing the normal signal.
So it stops the biogenic amines from working properly.
Essentially, yes. They compared bees treated with 3 micrograms of cocaine alone versus bees treated with 3 micrograms of cocaine plus 2 micrograms of mianserin applied together.
And they knew manserin itself wouldn't just make the bees sick.
Good point. They had previously established that this specific dose of mianserin doesn't make bees sick or reduce dance performance on its own. But it is an effective antagonist at blocking those amine receptors.
So the idea here was really elegant. If manserin, which blocks these pathways, could counteract the effects of cocaine.
It would strongly suggest that cocaine was indeed acting through those very same biogenic amine pathways, just like it does in mammals.
So they weren't just looking at behavior, they were trying to pin down the underlying neural mechanisms right from the start. And they were really trying to differentiate between cocaine just stimulating motor pathways, making them twitchy,
Right.
versus actually changing the bee's perception of reward, correct?
You've hit the nail right on the head. That was a central challenge. The honeybee dance, while it's communication, is fundamentally a motor behavior. It involves movement.
So maybe cocaine just makes them move more, generally.
That was the critical alternative explanation they had to rule out. The possibility that cocaine was simply causing general motor stimulation, making the bees hyperactive or twitchier in a non-specific way, which could look like more dancing, but wouldn't actually mean they perceived more reward.
That distinction is crucial. And that led directly to experiment 2, right? To isolate those potential motor effects.
Precisely. Experiment 2 was designed specifically for that purpose, to test if cocaine generally increased locomotor behavior. If it did, then maybe the increased dancing was just a byproduct of general motor stimulation, not related to reward.
So how did they go about isolating those potential motor effects? How do you test if a bee is just generally more active because of the drug outside the hive context?
They caught forager bees, the same type used before. They briefly chilled them, just for about 2 minutes at minus 20 degrees Celsius, just to make them hold still for a moment.
Quick chill, okay, for handling.
Just long enough for handling. Then they applied either 3 micrograms of cocaine, that lowest effective dose from experiment 1, or just the DMF solvent as a control, topically to their backs, just like before.
And then what? Put them somewhere to watch them move.
Exactly. Each treated bee was then placed individually into a standard 9-centimeter Petri dish. Nothing fancy, just a clear plastic dish. But the bottom of the dish had a simple radial grid drawn on it, like spokes on a wheel.
Okay.
They then meticulously recorded the number of times the bee walked across one of those lines on the grid during a 5-minute observation period. This movement index gave them a quantifiable measure of the bee's general activity level. More line crossings equals more movement.
Simple but effective. And they observed them more than once.
Yes, each bee was observed twice, once at 30 minutes after treatment and again at 60 minutes post-treatment. This timing matched the period when they were seeing dance effects in Experiment 1. And this kind of assay had been used successfully before to detect activity differences in treated bees.
Okay, so if the bees were just generally hyperactive or twitchy from the cocaine, you'd expect them to cross significantly more lines in that dish compared to the control bees.
That was the prediction if it was just general motor stimulation.
Got it. What about Experiment 3? How did they assess the bees' reward sensitivity in a completely different way, totally outside the context of dancing?
That was the precise objective of Experiment 3, to determine if cocaine increased responsiveness to rewarding stimuli using a well-established standard lab technique called the proboscis extension response assay, or PER assay.
PER assay? How does that work? Do you have to restrain the bees?
You do. For this, individual bees were carefully harnessed in tiny metal tubes. It sounds weird, but it's a common and humane laboratory method. It restricts their body movement, but leaves their head and important sensory organs, like their antenna and proboscis, free and accessible.
Okay, so they're held gently but firmly.
Right. Then they were starved for 4 hours beforehand. This doesn't harm them, but it makes sure they're motivated to respond to sugar when it's presented.
Makes sense. Makes them hungry.
Then, 1 hour before the actual testing began, they were treated topically, again on the back, with either 3 micrograms of cocaine in DMF, or with DMF only, or they were sham treated, just touched, as the controls.
Same treatments as before. And how exactly does this PER assay measure reward sensitivity? What are they looking for?
It's quite elegant in its simplicity. They present tiny 20 microliter drops of 7 different sugar solutions to the bee's antenna.
Okay.
These solutions systematically range from very, very dilute, like 0.1% sucrose, barely sweet, all the way up to very concentrated, 60% sucrose, and even pure honey. They're presented in ascending order of concentration, from weakest to strongest.
And what happens when the sugar touches their antenna?
If the bee perceives the concentration as rewarding enough, it will reflexively extend its proboscis, that long straw-like mouth part it uses for drinking nectar. It's an involuntary feeding initiation response.
Ah, like it's getting ready to drink.
Exactly. And the measurement they take is the sucrose sensitivity index. This is simply the first concentration in that ascending series that reliably causes the bee to extend its proboscis.
So a lower index means they respond to weaker sugar solutions.
Precisely. A lower index means a higher sensitivity to sugar. It indicates that even a barely sweet solution is enough to trigger their feeding response, suggesting they perceive it as more rewarding.
Clever. And they controlled for just general sensitivity.
Yes, critically, a drop of plain water was presented between each sucrose presentation, and they carefully excluded any bees from the analysis that responded to the water more than twice.
Why exclude them?
Because responding frequently to water would indicate a non-specific general sensitization, maybe just reacting to anything touching their antenna, or a generalized reflex hypersensitivity, rather than a specific increased response to the sucrose reward itself. This helped ensure they were truly measuring reward sensitivity, not just general twitchiness.
Okay, so this gave them a completely different angle on how cocaine might affect the bee's perception of reward, moving beyond just the dance behavior. But they didn't stop there, right? Experiment 4 looked beyond sugar.
That's right. They wanted to know if this effect was specific to sugar or if it applied to other important resources bees collect. Experiment 4 was designed to test if cocaine affected dances for resources other than just ingested sucrose.
Like pollen.
Exactly like pollen. This time, the resource offered at the feeder, again 10 meters from the hive, was freeze-dried pollen presented in a dish.
And pollen is different because they don't eat it right away.
Right. Pollen is a crucial protein and lipid source for the colony, vital for feeding larvae. But unlike nectar, which the foragers ingest directly, pollen is collected in specialized baskets on their hind legs and carried back to the hive. The foragers themselves don't typically ingest it during the foraging trip.
So it's a different kind of reward, valuable but collected, not eaten immediately.
Exactly, a different modality. Again, individually marked bees were treated with 3 micrograms of cocaine or the sham DMF controls.
And what did they measure here? Still dance rate?
Well, Pollen dances are naturally rarer and often shorter than nectar dances, especially during the time of year they did the experiment late autumn in Canberra. So measuring rate wasn't as practical. Instead, they measured a different metric: the proportion of bees that performed at least one dance after returning from the pollen dish during a 50-minute observation period, starting 20 minutes post-treatment.
So basically, did the cocaine make them more likely to bother dancing for pollen at all?
That was the question. This was vital to see if cocaine's effect was generalized to different types of floral rewards, not just boosting the response to injected sugar. Was it a broader effect on reward assessment?
Makes sense. Okay, that covers experiments 1 through 4. And finally, experiment 5. This is the one that sounds conceptually wild: chronic cocaine and looking for withdrawal in bees. That seems like a huge leap.
It truly is audacious, but also incredibly insightful scientifically. The objective here was precisely to examine if bees would exhibit a withdrawal-like response. Could they model something akin to the cognitive deficit seen during withdrawal in human cocaine users or in rat models?
That's a big ask for a bee study. How did they even approach it?
It required a long-term exposure setup. They started with newly emerged adult bees. These bees were reared from emergence in controlled groups, about 60 bees per cage, kept in a humidified incubator at a constant warm temperature, 32 degrees Celsius, for 6 continuous days.
So raised in the lab from day 1.
Right. And during this entire 6-day period, they received chronic oral drug treatments. This time, the drug wasn't applied topically, it was mixed into their food.
What food?
They were fed ad libitum, meaning they had constant access whenever they wanted to sugar solutions, 1.5 mol sucrose, that contained either a non-toxic dose of cocaine hydrochloride, 0.66 milliball, or octopamine hydrochloride, 10.54 millimol, or just the plain sucrose as a control.
So 3 groups: chronic cocaine, chronic octopamine, and control, all drinking it for 6 days.
Correct. Continuously exposed. Then on day 6, after all that chronic exposure, they tested their learning ability.
How did they test learning? Another PER assay.
Yes, they used a well-established two-odor discriminant learning task, again employing the PER assay, which is a standard method for testing associative learning and memory in bees.
Okay, associating smells with outcomes.
Exactly. They used commercial lemon essence dissolved in a rewarding 1 ml sucrose solution as the good stimulus, CS+, and natural vanilla essence dissolved in a non-rewarding salty saturated NaCl solution as the bad stimulus.
Lemon means sugar, vanilla means salt. Got it.
The bees were given 3 distinct training sessions, spaced 10 minutes apart. During training for the lemon odor, when the smell was presented, their antenna was gently touched with the associated sugar stimulus. This triggers the PER, they taste the sugar, and ideally form an association. Lemon smell predicts sugar.
And for vanilla?
Same procedure. Vanilla smell presented, antenna touched, but this time with the unpleasant salt solution. So vanilla smell predicts yucky salt.
Okay, standard conditioning. Learn the difference.
Right. Then comes the memory test. 20 hours after this training, their responses to just the vanilla and lemon odors alone were tested. A correct response was defined as extending the proboscis, PER, to the lemon odor, but not extending it to the vanilla odor. This shows they learned and remembered the discrimination.
Makes sense. So where does the withdrawal part come in? How did they test that specifically?
This is the truly clever part of the design. During that 20-hour interval between the training and the final memory testing, the cocaine and OA-treated bees were divided into 2 crucial subgroups.
Okay.
The first subgroup was the withdrawal group. These bees were fed small amounts, 20 microliters, of plain sucrose solution at 1, 3, and 7 hours after the training ended. The critical point is this ensured they had a full 20 hours without any further oral drug exposure before the final memory test. They were taken off the drug.
So they went into withdrawal, potentially?
That was the hypothesis. The second subgroup was the chronic group. These bees, in contrast, continued to receive small feedings of the drug-containing sucrose, either cocaine or OA, at those same 1, 3, and 7-hour time points after training. So they maintained consistent drug treatment right up until the memory test.
So one group stayed on the drug, the other group stopped getting it.
Exactly. And the sucrose control group, of course, was fed plain sucrose in both conditions, during the chronic phase and during the 20-hour interval.
Wow. So by comparing the learning performance on the memory test between the chronic group, still on the drug, and the withdrawal group, drug stopped,
they could isolate and observe any potential cognitive effects specifically caused by the cessation of chronic cocaine exposure.
That's a truly sophisticated design for an insect study. It really allows them to ask, does stopping the drug mess up their memory?
It really is ingenious. By meticulously comparing learning performance, normalized to the sucrose control group, across these two conditions, continuous drug versus drug cessation, they could assess not only the effects of chronic treatment itself, but critically, the effect of abrupt cessation. This meticulous design let them dissect the bees' behavioral and cognitive responses to cocaine with unprecedented detail.
Okay, so we've covered the compelling why, the paradox, the neurobiology, and the
Exactly. The more cocaine they received, within that tested range, 3, 6, 12 micrograms, the more likely those individual foragers were to perform a round dance when they got back to the hive. And if they danced, their dances were more vigorous, faster, more circuits per minute, lasting longer.
wow. So it wasn't just random movement. This is specifically the communication dance being amplified.
Precisely. And importantly, this is exactly how dance behavior would be predicted to change if the foragers were reporting resources of greater value than they actually were.
It was as if the cocaine made them overestimate the quality or abundance of the sugar water they just found. Like they were exaggerating how good it was.
That's the interpretation, yes. It looked just like they'd found a much richer nectar source.
So it wasn't just random hyperactivity or twitching. It was a specific, meaningful form of communication that was amplified, almost exaggerated. And you mentioned octopamine, the positive control. Earlier, was there a clear parallel between cocaine's effect and OA's effect on dancing?
Indeed, the parallel was striking. The effects of cocaine on dance behavior were remarkably similar to those caused by octopamine. And remember, OA is already known to modulate reward processing and increase dancing in insects.
So cocaine was acting like a known reward-related chemical in bees.
The similarity in the behavioral response was immediate and strongly suggested a common underlying mechanism involving those biogenic amines.
And speaking of mechanism, what about that sub-experiment with miancerin, the blocker? Did that work?
It provided crucial confirmation. When bees were simultaneously treated with miancerin along with the cocaine, the effects of cocaine on their dance behavior were almost completely eliminated. The dancing went back down to control levels.
Wow. So blocking the biogenic amine pathways stopped cocaine from boosting the dance.
Exactly. This powerfully suggests that cocaine modulated dance performance by directly interfering with or acting upon these biogenic amine signaling pathways, much like it does in mammals. This was a huge piece of the puzzle, tying the behavioral effect directly to a specific neurochemical system.
That's a critical link right there. But what about their general foraging ability? Were these cocaine-treated bees just hyped up and dysfunctional, or could they still function as effective foragers?
That's a crucial point. The study meticulously monitored the bees, and it found that cocaine-treated bees still foraged normally and effectively between their dance bouts.
So they could still fly. Okay, find the feeder, drink the sugar.
Yes, they were perfectly able to fly, locate the feeder, collect the sucrose, and return to the hive without any apparent impairment in their basic motor skills or foraging efficiency at these doses. This indicated that the doses used didn't cause general motor dysfunction or incapacitation. They weren't just wrecked.
That's important. And another critical point you mentioned earlier that reinforces the reward idea rather than just motor stimulation.
Right. Cocaine only stimulated dancing when bees were in the appropriate social context. That is, when they were back in the hive, on the dance floor area of the comb, surrounded by potential recruits.
What about bees treated but kept alone?
Bees treated with cocaine but then immediately isolated from the hive never performed dances.
Ah, so it wasn't a simple, aimless motor release like a muscle spasm. The behavior still required its natural social trigger and context?
Exactly. The dance retained its meaning and its function as communication. It wasn't just random movement.
So the dancing wasn't just aimless twitching, it maintained its context and communicative purpose, just amplified. That leads us right into experiment 2, where they specifically looked for that general motor hyperactivity outside the social context of the hive. What did they find there? Did cocaine make isolated bees run around like crazy?
The key finding from experiment 2 was absolutely definitive. No. There was no sign whatsoever of motor malfunction or general locomotor hyperactivity in those isolated bees treated with the 3 microgram cocaine dose.
Really? Not at all.
When placed in those petri dishes with the grid, they moved across the lines at statistically the same rate as the control bees, the ones treated with just DMF. They weren't jittery, they weren't uncontrollably active, they weren't spinning in circles. Their general movement level was normal.
And why is that result so critically important? Why was it so essential to rule out general motor stimulation?
Because it argues strongly and unequivocally against that alternative hypothesis, the idea that cocaine was merely stimulating general motor pathways, which could have been a simpler, non-reward related explanation for the increased dancing.
If it just made them twitchy, they'd dance more.
Right. If the increased dancing seen in experiment 1 was just due to generalized hyperactivity, you'd expect their movements in the petri dish to be random, uncontrolled, maybe faster, regardless of context. But that's not what they saw.
Instead, the dancing was context-dependent, socially triggered, and this experiment confirmed there wasn't a non-specific motor activation happening at these doses.
Precisely. It showed that the increased dancing wasn't just aimless twitching. It was a targeted, meaningful behavior, amplified, presumably, by how the bee perceived the value of the resource it was communicating about.
Okay, so definitely not just general motor stimulation. What about experiment 3, the proboscis extension response, the PER assay? This tested sucrose responsiveness in a very different way. Did cocaine make them more sensitive to sweetness itself?
Here, the key finding was a resounding yes. Cocaine significantly increased responsiveness to sucrose in the PER assay.
How significantly?
Bees treated with cocaine extended their proboscis at lower sucrose concentrations compared to the control bees. Their sucrose sensitivity index was lower, meaning they reacted to weaker sugar solutions.
So it was like they were saying, even this barely sweet solution tastes amazing.
That's a good way to put it. They required less sweetness to trigger that feeding initiation response. And importantly, the study also confirmed the specificity of this. Cocaine did not increase the number of bees that non-specifically sensitized to the plain water presentations between sugar trials.
So it wasn't just a general heightened sensitivity to anything touching their antenna or a non-specific reflex getting triggered more easily. It was specific to the sugar reward.
Precisely. This strongly suggests that cocaine specifically increased responsiveness to sucrose reward, rather than causing some kind of general sensitization of reflex motor responses to any antennal stimulation. It directly reinforces the idea that cocaine was modulating their perception of reward, making the sugar appear inherently more appealing or valuable to the bees, even at very low concentrations. It's like it turned up the deliciousness dial biochemically.
That's fascinating. And they didn't just stop at sucrose, did they? Bees collect more than just nectar. Pollen is crucial too. So experiment 4 looked at dances for pollen. Did cocaine affect that too? Did it broaden the scope of what it was amplifying?
Yes, it did. The effect wasn't limited to sugar. The key finding from experiment 4 was that cocaine also significantly increased the proportion of bees that performed at least one dance after returning from the pollen feeder.
How big was the increase? You said pollen dances were rarer.
They were. In the control groups, only about 22% of untreated bees and 27% of DMF-treated bees danced for pollen during the observation period. But among the bees treated with 3 micrograms of cocaine, a much higher proportion, 45.6%, performed at least one pollen dance.
Wow, that's almost double the rate of the controls. That's a substantial increase.
It was a statistically significant and biologically meaningful difference, yes.
What does that imply about the scope of cocaine's effect? Is it just about taste?
It demonstrates powerfully that cocaine modulates responses to collected resources other than just ingested sucrose. It indicates a more general effect on floral rewards encompassing both nectar, ingested sugar, and pollen, collected protein lipid. This further supports the idea that cocaine impacts the general reward assessment system in the bee brain, rather than just acting on a specific sensory pathway for sugar taste.
So it wasn't just making sweet things sweeter, it was making the overall value of collected resources, regardless of their specific type, seem higher and more worthy of communication through dance.
That appears to be the case. It suggests a broader modulation of how the bee evaluates the goodness of what it finds out in the world.
Okay. And finally, the big one, experiment 5, chronic cocaine treatment, and what they daringly called the withdrawal phenomenon. This is where it really gets mind-bending, connecting bee behavior to concepts usually reserved for mammals. What were the results here? Did they see withdrawal?
This was arguably the most profound and unexpected finding of the entire study. Let's break it down carefully. First, let's look at the learning performance in the chronic treatment group, the bees that stayed on the drug the whole time.
Okay, the ones tested while still receiving cocaine or OA?
Right. Their learning performance in that two-odor discrimination task did not differ from the untreated control bees or the OA-treated controls if the drug treatments were maintained continuously during that 20-hour period between training and testing.
So being on cocaine continuously didn't mess up their ability to learn the task?
Correct. This is crucial because it showed that continuous cocaine exposure at these doses didn't impair their fundamental learning ability. They could learn the lemon good, vanilla bad association just as well as control bees. This argues against the drug just causing general toxicity or chronic cognitive impairment that would prevent learning.
Okay, so as long as the drug was present, their learning was normal. But the critical result came from the other group, the ones where the drug was removed, correct? The withdrawal group.
Precisely. The truly groundbreaking result came from that withdrawal group. If cocaine, or interestingly octopamine 2, treatment ceased during that 20-hour interval between training and testing, meaning they were abruptly taken off the drug after being on it chronically.
What happened to their memory?
The cocaine-treated group performed only half as well on the memory test compared to the control group or the OA-treated withdrawal group. Their learning score, their ability to correctly remember which smell predicted reward, plummeted by 50% only when the drug was withdrawn.
Whoa. So the learning deficit only appeared when the drug was taken away, after chronic exposure, not when it was consistently present. That sounds strikingly similar to cognitive deficits seen in mammalian models of addiction withdrawal.
Absolutely. It's a significant and undeniable learning deficit that manifests only upon the abrupt cessation of prolonged cocaine exposure. This is strongly, powerfully suggestive of a withdrawal-like phenomenon in bees.
And it wasn't just that they were sick from stopping the drug.
It's highly unlikely to be due to general sickness because, as we just noted, the chronic group, which was still on cocaine, performed normally. If stopping the drug just made them generally ill and unable to perform, you might expect the chronic group, still getting the drug, to also be somewhat impaired, but they weren't.
Wow. So this is the first time something like this had been seen in insects.
Unequivocally, yes. The first time such a specific cognitive deficit, appearing only upon drug cessation after chronic exposure, something so analogous to withdrawal effects, had ever been demonstrated in insects. It's a truly groundbreaking parallel to mammalian responses and offers a whole new way to think about how broadly these fundamental neurobiological mechanisms related to drug effects might be conserved across the animal kingdom.
Okay, let's try to put all of this incredible detail together. We've got bees dancing more vigorously and more often for both sugar and pollen after getting cocaine.
Sure.
We've got them becoming more sensitive to even weak sugar solutions, suggesting they perceive it as more rewarding.
Yep.
We know it's not just general motor stimulation because they aren't hyperactive when isolated.
Right, crucial control.
And then this bombshell finding. After chronic exposure, stopping the cocaine causes a significant drop in learning performance, looking an awful lot like withdrawal.
Check. Mind-bending stuff.
So when we put all these pieces together, what does this all truly tell us? What's the bigger picture here? What did Barron and colleagues conclude?
What's truly fascinating here, and what they concluded, is how these findings compel us to fundamentally rethink that long-held assumption about drug reward. The idea that insects just react negatively. The sum of the evidence really converges powerfully.
Meaning?
The increased dance for both resources, the enhanced sucrose responsiveness, and especially that dramatic withdrawal effect on learning, all strongly support the interpretation that cocaine directly modulates reward processing systems in the insect brain. It's not just disrupting motor control, it's tapping into their value assessment circuits.
And this was the first direct evidence for that in insects.
This was without a doubt the first direct, unambiguous evidence of such a reward-modulating effect in insects. It's a genuine game changer for understanding comparative neurobiology and the evolution of reward.
And it really robustly rejects the idea that cocaine was merely stimulating their motor systems, doesn't it? Because that would have been the easier explanation, the one that kept the old mammals versus insects paradigm intact.
Absolutely. The researchers went to extraordinary lengths, as we've detailed, to meticulously exclude that motor-only hypothesis.
Experiment 2, the locomotion test.
Right, showing no general hyperactivity. And experiment 3's PER assay showing specificity to sucrose reward, not just general sensitization. Plus, remember the social context dependence of the dancing itself. The fact that bees only danced when they were actually back in the hive, communicating.
Right. It wasn't just some aimless twitching or a simple motor stereotypy. It retained its full meaning and social context. It was the value signal that was being amplified.
Exactly. The behavior was qualitatively normal, just quantitatively enhanced, suggesting the underlying motivation or perceived value was altered.
So it's not just that bees react to cocaine, but that they seem to process it through a reward system that has these striking neurochemical and behavioral parallels to our own. Let's deep dive into the underlying mechanism again, the biogenic amamine story. That meandering result was key.
Indeed. The fact that meandrin, the known biogenic amine antagonist, effectively eliminated cocaine's dance-enhancing effects in Experiment 1 is the crucial piece of the mechanistic puzzle.
Because meandrin blocks those pathways.
Right. Since meandrin interferes with biogenic amine signaling, likely including dopamine, octopamine, and serotonin receptors in bees, its ability to block cocaine's effect powerfully implies that cocaine influences bee behavior by directly interacting with these specific biogenic amine pathways.
Which is exactly what it does in mammals.
It's entirely consistent with its primary known mode of action in mammals, where it blocks the reuptake transporters of these menamines, most notably dopamine, leading to increased signaling. It's like finding the same key fitting the same type of lock in two vastly different houses, a beehive and a human brain.
So essentially, cocaine is messing with the same fundamental chemical communication systems, the same basic neurobiological machinery in both bees and mammals. That's a profound thought about evolution.
It truly is. And it wasn't entirely out of the blue, mechanistically. We already knew that cocaine-sensitive dopamine and serotonin transporters had been identified and characterized in Drosophila, the fruit fly.
Okay. Another insect.
And manipulating these specific systems in flies had been shown to directly affect their sensitivity to cocaine's motor effects. Some other insects, like the cabbage looper moth, were even known to have distinct transporters for octopamine, dopamine, and serotonin that could potentially be targets.
So the basic components were known to exist in insects.
The parallels at the molecular level are surprisingly strong, indicating a deep evolutionary conservation of these basic signaling systems going way, way back. Now, it's important to acknowledge, as the authors did,
Yes.
that while octopamine has been clearly implicated in bee reward processing and learning from previous work, the specific biogenic amine transporters present in honeybees and their precise molecular sensitivity to cocaine were still uncharacterized at the time of this study in 2008.
Ah, so they knew the system was involved, but not the exact molecular handshake yet.
Precisely. While the pathway seemed clear, biogenic amines, the exact molecular targets within the bee brain, and how strongly cocaine bound to them, still represented an open and exciting question for future research. But the functional evidence was compelling.
Okay, so if we connect all of this back to the bigger picture, this study isn't just about the fascinating drug responses of honeybees. It proposes a fundamentally alternative solution to the paradox of cocaine reinforcement, a much more elegant, what scientists call a parsimonious explanation. Simpler, fits the facts better.
That's the beauty of it. It provides a way to resolve the paradox without having to assume completely different brain mechanisms. It doesn't deny cocaine's role as a plant defense. Cocaine is an incredibly effective plant defense because, as a neurotoxin, it causes catastrophic failure of insect motor control at high doses by disrupting those vital biogenic amine signaling systems.
Okay, so the deterrence part holds true. It does poison insects badly.
That part holds true. That's why it's a good deterrent when an insect tries to eat a lot of the leaf. However, here's the elegant twist proposed by this research. Because the very same biogenic amine systems that regulate critical motor function also happen to modulate crucial reward processing.
In both insects, via dopamine and octopamine, and mammals, primarily dopamine.
Right. Because of this shared dual function, it is, in a profound biological sense, almost unavoidable that a compound like cocaine, which targets these systems, would also impact reward systems, at least at certain doses.
So the reinforcing, even addictive properties of cocaine in mammals and potentially the reward-like effects seen in bees at low doses are almost like an accidental side effect of targeting a system with multiple jobs.
That's the idea. The coca plant didn't evolve cocaine to be rewarding to animals. It evolved it to deter them, to poison them by messing up their movement. But the shared neural machinery means that targeting that machinery for deterrence comes with an unintended, almost inescapable consequence for reward pathways in certain organisms, including us, and it seems, including bees.
It's a classic example of evolution being thrifty, maybe. Using the same system for different things, leading to unexpected outcomes when something interferes with it.
You could certainly see it that way. Evolutionary repurposing or conserved function, leading to pleiotropic effects, where targeting one function inadvertently affects another linked one.
And why does it still work so effectively for the plant as a defense in nature if it has this potential reward effect?
Because, as the researchers point out, the natural concentrations of cocaine found in coca leaves are extraordinarily high. So high that herbivorous insects, when they try to feed on the plant, very rapidly ingest a toxic, incapacitating dose.
Ah, so they get the nasty motor effects way before any subtle reward effect could matter.
Exactly. At those high, naturally occurring, incapacitating concentrations, any potential reinforcing properties become utterly irrelevant to the plant's defense strategy. The insect is already experiencing severe motor dysfunction, is strongly deterred, and quickly leaves the plant or succumbs to the toxicity. So the plant's defense strategy remains highly effective in the real world, despite this fascinating side effect revealed by low-dose lab experiments.
This truly raises an important, even profound question for you, our listener, to consider. What does this startling discovery imply about the universality and deep evolutionary conservation of certain neurochemical systems across the entire tree of life?
Indeed. Think about it. If such complex behavioral and cognitive responses to a specific chemical, a plant toxin, no lesser share between organisms as seemingly disparate, as incredibly far apart on the evolutionary tree as humans and honeybees.
Millions and millions of years of separate evolution.
What does that truly tell us about the fundamental, ancient building blocks of reward, motivation, and maybe even addiction in the animal kingdom? It suggests an incredibly deep evolutionary conservation of these basic neural mechanisms.
Hinting that some of the most fundamental processes in our own brains, things we think of as uniquely human or mammalian, might be rooted in biology that predates even the appearance of mammals, maybe even vertebrates.
That's the mind-bending implication. What really stands out to you listening to this about how this study so meticulously and ingeniously dissected such a complex behavioral and neurochemical question using bees and their dance?
This deep dive has really shown us that the paradox of drug reward might not be a paradox at all, but rather a fascinating, perhaps even inevitable, consequence of evolution's incredible yet sometimes surprising reuse of fundamental biological machinery.
It's elegant in a way.
The shared biogenic amine systems between a tiny honeybee and a complex human mean that a plant's defense mechanism, evolved for one purpose, deterrence, can have utterly unexpected side effects that resonate across vastly different species, tapping into the very core of what drives behavior. Reward.
It's a powerful reminder that even in organisms we might perceive as simple, the intricate dance of neurochemistry can mirror our own complex inner worlds in surprising ways.
So for our final provocative thought for you to chew on today, if a plant's defense mechanism can serendipitously tap into the very reward circuits of a honeybee, and by extension ourselves, what other complex behaviors or chemical responses out there in the natural world might also be the side effects, evolutionary adaptations that we haven't even considered yet?
That's a great question. With seemingly unrelated phenomena, maybe social bonding, maybe foraging strategies, maybe responses to stress, could share a deep biological root, a conserved mechanism that's just waiting to be uncovered by the right experiment.
It really makes you wonder how many other hidden commonalities exist just under the surface, connecting all forms of life, just waiting for a deep dive.